Meet Toxoplasma gondii, a unicellular pestilent protozoan with nefarious tendencies. This parasite uses creative tactics to torment and pervade a range of hosts and ecosystems worldwide. It's one of the most common parasites in humans, infecting about a third of the global population. But its main target is cats. Obsessed with all felines of any size, it infiltrates the organisms surrounding feline populations to promote its evil plan of world domination. T. gondii has a complex life cycle involving multiple hosts and infectious stages. This microscopic menace has one goal in life: reproduce and multiply. Its days and nights are spent thinking about how to find its way back to its definitive host. The gut of a cat is the only place T. gondii can reproduce sexually and form oocysts. These oocysts are T. gondii's method of spreading mayhem. Once inside El Gato and successfully multiplied, the feline sheds these oocysts by pooping them out. Once they've been shed in feline feces, they make their way into the waterways and infect any warm-blooded animal who unwittingly quenches their thirst with the contaminated water. While farm animals and other rural creatures can become infected, cats are also the primary conduit in dense populations of human metropolises. And even though cats are the definitive host of interest, rats seem to be the most often the warm-blooded animal carriers of choice, and for good reason. Because these protozoans are non-motile, it relies on its victim's movement near and around the holy grail of hosts, cats, and it doesn't leave this movement up to chance. It employs mind control techniques to alter the behavior of what it considers a far inferior species. Once rats ingest the parasite, T. gondii alters the rat's natural fear mechanism by focusing its cyst production in the amygdala of the brain, rewiring the rodent's natural fear of cats into an obsessive need to be near them. The instinctual aversion to the scent of cats in order to avoid being eaten is transformed into an intense attraction. Uninhibited by survival instincts, the intoxicated rodent offers itself up to El Gato. To this microscopic menace, these vermin are merely collateral damage, a means to an end. They exist only to be manipulated in service of the survival and transportation of the maniacal T. gondii. T. gondii is not only a host-jumping, mind-controlling menace, it's also a sneaky shapeshifter. This parasite has a complex reproductive life cycle, which allows it to evade host defenses, survive in harsh hostless environments, and proliferate in optimal conditions. If we were to take a microscopic look, we'd be able to see the complexity of each. Depending on the host and the phase of reproduction for which it's optimized, T. gondii changes from unsporulated oocysts to sporulated oocysts in the environment to tissue cysts, which are tachyzoites, which is the infective stage in which the cyst rapidly reproduces many crescent-shaped cells and intermediary hosts, and then it forms into bradyzoites, which is the slower reproduction phase of the infective tissue cysts. Inside the intestine of the cat, T. gondii forms gametes which fuse to create zygotes and then undergoes meiosis. Once fertilized, an oocyst wall is formed around the parasite. The cat's infected epithelial cells rupture and then discharge oocysts into the intestine. The cat poops and the cycle starts all over again. The oocysts are patient agents of T. gondii's agenda. They can sit in the environment for months or years waiting to be ingested by their next victim. Once inside a human host, this parasite can cause a range of symptoms, severity, and damage, sometimes after living years dormant lodged in the central nervous system, lymphatic system, and the brain. The resulting infectious disease is called toxoplasmosis. While infection is common and most people who are infected are asymptomatic, disease cases are less common and include flu-like symptoms for adults with properly working immune systems. It can cause more serious issues for people who have compromised immune systems and infected fetuses who have contracted from their mother through the placenta. For babies with congenital toxoplasmosis, they can exhibit developmental issues, intellectual disabilities, seizures, and vision problems. While being around cats is a primary risk factor for infection for any animal species, humans can also be exposed to the pestilent protozoan by eating undercooked or raw meat or exposure to contaminated soil or water. When T. gondii makes its way inside our bodies, it first triggers our immune system, causing the parasite to produce cysts. When the invasion happens in body with a suppressed immune system, the effects of the infection are harsher on its host. Instead of producing cysts and laying in wait, T. gondii starts to proliferate rapidly, causing severe disease. More vulnerable populations can experience attacks on the central nervous system, brain, lungs, and heart, with symptoms including fever, seizures, psychosis, and then problems with vision, speech, and movement. T. gondii is part of the Protus kingdom in the fifth phylum known as Apicomplexa. This lineage of sporozoa shares similar affinities for disease-causing activities. These protozoa are non-motile and share a structural characteristic known as the apical complex, which helps aid their invasion to hosts. Some notable relatives are the agents of malaria, tick-borne diseases, and food and waterborne intestinal infections. While we have a good understanding of T. gondii's partiality for the brain and its fear-altering effects on rodents, we don't yet have the full picture on the mechanism of influence on human behavior. There are observational and metadata studies that point toward a similar association between T. gondii infection and psychological disorders, as well as more subtle shifts in behavior, like risk-taking and fear inhibitions. Researchers have found higher rates of toxoplasmosis infection in populations of people with active psychosis and schizophrenia. Dormant toxoplasmosa has only until recently been considered a relatively benign infection. Undoubtedly, scientists will continue to observe and uncover the covert actions of this pervasive parasite to hopefully understand more about what its intentions are in our brains.